Okay. Do you see my screen?
Yeah, we see it.
Okay. So let's start. So chapter 11, it talks about mechanism of microbial genetic. So basically we are going to work on the DNA replication and how DNA actually responsible to make proteins. Okay, so mostly we are going to work on the functions of DNA. Okay, so DNA, the genetic material. So DNA has two functions. So if we write it down, the function, oh, my Wacom tablet is not working today properly here. So function of DNA, it has two functions. Okay, the first one, this is actually responsible for the inheritance of genetic material. So hereditary material, it passed down the genetic information from parent to offspring. Okay, so that is one function of DNA and very important function. Okay. The second function of DNA is it actually directs the protein synthesis. And that making protein synthesis is very important for our growth and development. So this chapter will talk about how basically the two functions of DNA. Okay, so we are going to learn the two functions of DNA in this chapter. The first part, the first function of DNA, which talks that it actually pass down the information from parent to offspring. So to do that, it needs to replicate. Okay, DNA needs to replicate. DNA needs to make its own copy. So in this chapter, we are going to learn how actually DNA replicate. And then the second function, that how DNA direct or control the protein synthesis procedure. Okay, so for the protein synthesis part, we have two processes, transcription and translation. Okay, so transcription and translation. So we are going to learn these two processes. So first we will work on the replication part, how DNA replicates, and then the second part, the protein synthesis, transcription and translation. And towards the end, very little portion, we are going to study about mutation. So that's all about this chapter. So the gene, what is gene? Gene actually, that's the portion of DNA, okay, or the chromosome that code for a specific trait. We talked about that on when we studied chapter 10. So if it is a DNA, remember that inside a DNA there are a few portions that actually code for a protein, means it's a coding sequence, and there are few portions where it does not actually make sense, means that's non-coding sequence. Okay, so that non-coding sequence is not actually the gene. This is not the gene, but the coding sequence. Coding means that carry the code for a protein. So basically cell can read and can understand the meaning of that code or coding. Okay, and cell directs that what or which protein body needs to make. Okay, so that coding means that. So gene is actually the portion of a DNA or chromosome that represent or that code for a protein. Now inside the DNA we have multiple coding sequence, means multiple genes are there, and we have 23 pairs of chromosome inside our body. So all these chromosomes, so 46 chromosomes, they actually carry that gene. So you can understand that millions of genes are there inside all of our chromosome. Together all these genes, so basically let's say this is our number 1 chromosome, probably in this chromosome we have maybe let's say 12,000 genes are there. So let's say 12,000 genes are there in this chromosome. So we have 46 chromosomes. So you can understand that if each chromosome carry, let's say 1,000 chromosomes, then 46 chromosomes. So basically that many genes are there inside our body. Okay, actually practically we have more than this number. Okay, so anyways, all these genes together, they are our genome. Okay, sum of all types is an organism's genotype. So all this genome means all these genes together, they make our genotype. Okay, and the expression of these genes is called phenotype. Okay, so now few things to clarify. Like as I told that each chromosome carry multiple of genes, but not all the genes are going to be expressed at the same time. Express means when actually body makes protein from a cell, and that protein synthesis, it's actually need based. Like let's say I need glucose at this moment. Okay, so not glucose, let's say I need insulin to control glucose. Okay, um, so that's the need at this moment. Body, what it will do, or the cell, you can say specifically cell, it will actually ask only the chromosome which code for that insulin. Okay, so only the gene for insulin will be expressed, means will be active that time, and ultimately it will help cell to make this protein. So basically based on the need, some genes, they become active. Okay, and all other remains inactive. So that's the reason we tell that not all the genes are going to be expressed at the same time. Okay, anyway, so when one gene is expressed, means that protein is going to be made. That protein represents the phenotype. Okay, means the external appearance of the body. So genotype means the genetic makeup. Phenotype is the expression of that genotype. But to remember that not all the gene is going to be expressed at the same time. Okay, all organisms contain more genes in their genotype than are manifested as a phenotype at a given time. Okay, as I told that not all the genes are going to be expressed at the same time. We have millions of genes inside our body, but maybe at this moment only one gene is going to make protein. That's the insulin gene. Okay. So phenotype is determined by the specific genes within a genotype that are expressed under specific condition. So now these genotypes, like the number of genes, that is actually fixed. Okay, that is constant. How many genes are there? That is actually constant. But I don't know, today my Wacom tablet is not working properly. Okay, so the number of genes are constant, but the phenotype, it actually varies based on some different factors like temperature, then acidity. Basically, environmental factors works on the gene. Like in lab, you can see that bacteria Serratia marcescens. Okay, that Serratia marcescens, if you culture the bacteria inside the incubator at 35 degrees centigrade, 35 to 37 degrees centigrade, it will make just white colony. Okay, but if you allow these bacteria to grow outside the incubator, like a normal temperature, like let's say 28 degrees centigrade to 30 degrees centigrade, then they make red colony. Okay, so over here, that for Serratia, gene expression, temperature has a big effect. Okay, and that the low temperature actually makes one red pigment. But if you increase the temperature, then that genes cannot be expressed. So environment has a big effect on the genotype. If you live in the tropical environmental conditions, or you can say in a warm temperature region, then your skin will be little more darker. But if you live in a cold temperature, cold weather, polar region, then your skin will be lighter. So that's the environmental effect. The same gene, like over here, that look like for the Serratia, same gene that actually responsible for the body color. Because of the different temperature, it expressed in different way, means it makes completely different protein. So in this case, I hope you understand that genotype remains constant, but due to the difference between the temperature, the temperature has an effect on that genotype. So basically, the phenotype changes based on the environmental condition. So genotype is constant, but phenotype is not. So flow of genetic information, how actually genetic information flows. So that's the first function of DNA. As I told, that first function of DNA is to pass down the genetic information from parent to offspring. So how it goes, okay? And I'm talking about the protein synthesis, so how this protein synthesis is related to the DNA. So if you can see the big picture, then it will be clear. So what happened? DNA. So first, let's say DNA. So DNA, what it does, so it has, remember that nitrogenous base, okay? So maybe adenine, guanine, thymine, cytosine, adenine, guanine, thymine, okay? So each three nitrogenous base actually represent one amino acids. And I hope you remember that amino acids are the monomer of protein. So multiple amino acids together, they make protein. Okay, so this is called triplet, or you can say that codon. When it's on the DNA, it's called triplet code. So that's how the DNA is actually associated with the protein synthesis. Okay, so over here, let's say this is the combination for my let's say number 1 chromosome. Okay, now when this is the combination, like this is amino acid 2, maybe this is the amino acid number 10, this is amino acid number 20. When these 3 amino acids are together, so basically number 2, number 10 and number 20 are together, then that will make one black pigment, so that my hair will be black. Okay, but instead of this combination, so for me it was 2, 10 and 20. Okay, number 2 amino acids, number 10 and number 20 amino acids. But instead of this combination, if you have let's say instead of 2 you have 14, then maybe 5 and 6 and then 7, then it could give you maybe let's say brown hair. Okay, because presence of these amino acids, okay, this combination actually is going to make one type of specific protein or pigment which is brown in color, and that will give your hair brown color. Okay, so that's how, I hope now you can understand how actually phenotype. So this is the phenotype, your hair is brown, that's the phenotype. Okay, but now that combination over here with amino acids, the composition of amino acids or sequences of amino acids, that is actually your genotype. Okay, so like over here, this is one codon, this is one codon, this is one codon, these are the genotype. But that sequence of this codon represent your phenotype. Okay, so when we tell that DNA is responsible to pass down the trait from parent to offspring, okay, what does it means? Means like this combination 2, 10 and 20. These combinations of or this sequence of amino acids on my chromosome number 1 that I actually got from my parent or from my dad or from my mom. Okay, and that gives my hair black color. So that's the genetic information. And this information actually starts from the DNA and ultimately ends up in a synthesis of a protein, very specific protein. And that's the flow of genetic information. Now it looks like pretty easy, like DNA is carrying the genetic sequence and body is making protein, but it's not that simple. Okay, there are 2 steps there. So if it is, so if it is a DNA, okay, so DNA is carrying all the genetic information, right? Now DNA is very precious because it is carrying all the genetic information, and DNA remains inside our nucleus over here. To make protein, the protein actually ribosome is responsible to make protein, and that ribosome remains in the cytoplasm. So how this information will come to ribosome? Okay, to bring that information all the way to the ribosome, then DNA has to come out of the nucleus. And that's very dangerous, because it can lose the end part, tail part of the chromosomes. Okay, it can be broken. So to avoid all these problems, what cell actually does inside the nucleus, um, it store all the DNA. It does not allow DNA to come out of the nucleus. Instead, what it does, a cell actually make a RNA copy of the DNA. Okay, and at this moment we can call that RNA as messenger RNA or mRNA. Now this mRNA, so it makes mRNA over here inside the nucleus. That mRNA comes out of the nucleus and goes to the ribosome. Okay, and so this is mRNA. Now ribosome can read that genetic information, that sequence, that 3 nitrogenous base at a time. Okay, and then following that instruction itself, ribosome actually makes protein. So it reads, okay, so this is number 2 amino acids, so it brings number 2 amino acids. Then the next one is number 10, so it makes that number 10. Then maybe number 20, so then it brings number 20. So all together, these three, when this is a sequence 2, 10, and 20, then it is going to make one protein. Maybe the name of the protein is, let's say, black protein. Okay, and that's how I'm going to get my hair black. Okay, so these are the 3 things happen. So basically, first step, DNA cell makes messenger RNA, or you can say RNA copy of the DNA, and this process is called transcription. Okay, so that's the transcript of DNA. And this process from messenger RNA, when ribosome make protein, that process is called translation. So genetic information is always goes from DNA to RNA to protein. Okay, so unidirectional. And this process, when genetic information moves from DNA to RNA to protein, by the process of transcription and translation, these two together is called central dogma of protein synthesis. Okay. So that's the central dogma of protein synthesis. So what is central dogma? So transcription and translation together is called the central dogma of protein synthesis, or central dogma of gene expression. Now, like when I told that DNA does not, cell does not allow DNA to come out of the nucleus, and this process is called transcription. So basically, it's like transcript. Like when you will graduate, what will happen? All of your grades and original documents will be here at GHC. But if you want to go to Kennesaw State University for higher study, so GHC is going to send your transcript over there, means the copy of the original version. Okay, if you want to apply to multiple universities, so GHC will send multiple copies of transcript. Okay, so that's why this process is called transcription. And then when it will go to Kennesaw, they will analyze. Okay, so this guy had studied biology, microbiology, so he can go to, he is allowed to study nursing or dental. So this analyzation, that's the translation. And another thing is here, until till on messenger RNA, it was actually cellular code. Okay, now when cell is making protein, that's the chemical code. So basically over here, the genetic code or genetic version of code is actually going to transfer into chemical form. So that's why this process is called translation. And together this transcription and translation is called central dogma. So now the DNA replication. Okay, so now we have idea that how actually DNA moves from the genetic information moves from DNA to RNA to protein. So DNA replication, the first function to perform its first function, DNA must replicate, because remember, so when one sperm fertilize one egg, okay, so together they make zygote, right? Two and one cell. Now that one cell actually multiply, divide, and then ultimately multiple cells are there inside a body of embryo, and ultimately that embryo grow and we become, say, adult, or from child to adult, all those developmental stages are there. So for this growth and developmental phases, so we started with, that sperm, it has that n number of chromosomes, only 23, and ova or egg had 23, so we started with 46 chromosome. So that only go on to one cell. Now we have trillion of cells. So how are we going to get all this genetic information? Remember that all of our cells need this all the genetic information. Okay, so that's why all of our cells are almost same. They carry the same genetic information. So that requires DNA replication. Now there are three different type of DNA replication. Okay, one is called the conservative process, semi-conservative, and dispersive process. So conservative process says that what happens, so DNA has a double helix structure, right? I hope you remember that it is made up with two strands. So in conservative process, what happens, the two strands, both the strands remain together, and they make a mirror copy of that strands, old strands. You can say this is the old one. So, and it's a copy of that old strands. So that's the conservative DNA replication process. Semi-conservative, it says that basically DNA cannot replicate when both the strands are together. Before replication, these two strands need to be separated from each other. And then each of the strand make their complementary copy. And that's how we get two DNA copies from one. So you can see that both the strands got first separated, and then they made complementary strands. So basically, when they get separated, the strands, they are the template strand. Okay, and they copy that template strand. So that's the semi-conservative. Dispersive says basically both the strands are both the copies of DNA. They are actually the combination of both old and new strands. So that's the dispersive. Now, which one is actually the correct way, or you can say that most of the organism follow which one? Okay, so now we know that most of the organism actually follow the semi-conservative model, but how do we know it? So like 52. That's the year Watson and Crick they gave us the they published the DNA structure, double helix structure. So you can imagine that when it is 1953, all the scientists, they put their money on that what type of DNA replication model actually we follow. So at that time everybody was busy to find out uh, what type of replication process we do. Michelson and Stahl they actually used bacteria E. coli as a model organism, okay, and what they did, they began growing that E. coli, Escherichia coli bacteria in a nutrient media or growth media, like that you use in lab, exactly same way. But only thing they changed that instead of normal regular nitrogen, they use the isotope of nitrogen. Isotope means basically like nitrogen, it has different isotope, means different version it has. Version means only the neutron, number of neutron varies from one to another version, okay? So they actually took two version or two isotope of nitrogen. One is called N14 version and another one is N15. So N15, inside N15, it has one more neutrons than the N14 one. So that N15 isotope is much heavier than N14. So they took two different growth media with one with N15 nitrogen and another one with N14 isotope. Okay, so first what they did, they actually allow all the bacteria to grow on N15, and then finally after that basically they allow bacteria to grow for multiple generation, okay, and when all the nitrogen, so basically when you grow the bacteria inside that growth media and it has that N15 nitrogen, to make DNA, bacteria will get that N15 nitrogen, right? Because that nitrogen is very important factor of DNA, okay? So to make DNA, they must get that N15 nitrogen from the growth media. So they allow bacteria to grow until all the nitrogenous bases of all DNA of those bacteria converts into N15 isotope. So basically, they allow bacteria to grow up to certain generations, and when so all the nitrogen base of DNA was N15 isotope. Okay, till then they were growing the bacteria inside this media. Now what happened? So if they do the centrifugation, means the spin the bacteria with basically it's a density gradient centrifugation. The name of the process is density gradient centrifugation. So the spin the bacteria with the very high speed. If they do that, this process basically it helps to make all the DNA a band. You can see that they separate, all the DNA gets separated based on their density. Okay, and then you can see the different bands based on the density. So so they did that in 15 after all the nitrogenous bases of those bacteria converts into N15, then they did that centrifugation and they saw that it's a pretty heavy isotope. So all the bacteria makes band over here towards the bottom of the centrifugation tube. Now what they did, they transfer all those bacteria who are carrying that in 15 DNA now. They transfer all those bacteria into another growth media carrying in 14 isotope. Okay. Now, when they spin, when they did that centrifugation, so what actually happened? You can understand. So it was in 15. That's the heavy one, and let's say this one. It's in 14, the lighter one. Okay. Now together, what will happen? They will make in between. So one strand will be in 14, one strand will be in 15. So together, they are actually in between in 15. So that's the N15 heavy one, and this is the N14. So the density of these two together should be here in between. Okay, so that's what it happens. This purple band, it's in between. So 14, that's very light, so that will create band over here, but the 15, it's heavier, so that will create band over here. But they noticed that when they transferred that N15 bacteria to N14 solution, it created the band which is in between N14 and N15, and that's the first generation. Okay, so so if it is conservative, then we could have found out two bands. After centrifugation, we could see two bands. Okay, but we just saw that there is only one band, and it's in between in 14 and in 15. So that actually reflects that either the replication process is semi-conservative or dispersive. Okay, now they look for second generation. So we had both red and blue. Okay, after second generation, we could found they noticed that there are two bands. So for dispersive, remember it is the portion. So few portion is in 15, few portion is in 14, and that should give us only one band. But instead, we noticed that two bands are there, because so one was the combination of two and another one was the red. So we found out two bands. So that actually proved that it is actually semi-conservative type of replication. And this experiment, it's a very famous experiment that proved that our DNA replication process follow the semi-conservative model. Now the DNA replication. Okay. So before we start the DNA replication, I hope you remember the structure of DNA. So few things I really need to go over again. So DNA are made up with the nucleotides, right? The nucleotides are the monomer, right? And all these monomer are attached, or you can say they are connected with each other by phosphodiester bond. Okay, now what's the structure of nucleotide? So nucleotide, it has sugar, inside it has nitrogenous base, and it has phosphate, right? So over here again, sugar, nitrogenous base, and phosphate. Sugar, nitrogenous base, phosphate. So these are three nucleotides. Okay, now if I draw the sugar like it is pentose sugar, right? And this is number 1 position, number 2 position, this is the 3, this is the 4th position, number 5 carbon is here, and phosphate is attached over here, and number 3 position we have 1 hydroxyl group, and nitrogenous base always remain attached with number 1 carbon, right? So what happens, so all these nucleotides remain attached. So we have, for these nucleotides, we have 3 OH position over here. So these 3 carbon and 5 carbon remain together. So if we see over this 3 carbon and this 5 carbon, they together make that phosphodiester bond, right? Okay. So I hope you remembered the structure. Now, on the other hand, if we notice the opposite side, so over here, nitrogenous bonds are over here, nitrogenous bases are here, then sugar, then phosphate. Okay, sugar, base, phosphate. So it goes like that way, like the other side. But in this case, what happened? So the sugar are like this way, because over here this should be number 1, and that's the nitrogenous base. So sorry, over here, and this should be the number 5 carbon over here, and this side should be 3 carbon. So basically, for this strand, it runs from, so this is 5, so this is 5 carbon. So this strand, one strand runs from 5 carbon, 5 to 3, but the other strand runs from 3 to sorry. Okay, so other strand runs from 3 to 3 to 5. Okay, these two are parallel, but they runs in opposite direction. So that's why these two strands are anti-parallel. Okay. So DNA replication. So and these two strands are together and they make a double helix structure, right? So like this. So if they are together, and remember it's semi-conservative model. So first thing what it has to do, that we have to separate the strands from each other. Okay, so that's the first step, that both the strands get separated from each other. And to do that, remember over here we have hydrogen bond. Okay, so we have to break that hydrogen bond to separate these two strands. Okay, so one enzyme, it's called helicase, that actually breaks this hydrogen bond. And as a result, two strands get separated from each other. And remember that these two strands, they are highly coiled, right? Because one chromosome, it's almost 3 to 6 meter long, and it has to fit inside a small nucleus. So they are really highly coiled and dense. Okay, so first thing, it has to be straight, so that cell can read, or the enzyme can read all the genotype sequence. We have another one enzyme, it is called topoisomerase enzyme. the topoisomerase, or you can say DNA gyrase. The DNA gyrase enzyme actually help to uncoil the DNA strand. Then another protein, another enzyme, it's called single-stranded binding protein. It gives weight. So it's like if it is a coil or spring, and if you put weight on the spring, then that weight actually help to stretch the spring, right? So if you add more weight over here, it will stretch more. So exactly that way, when it's a coiled structure, that single stranded binding protein put weight over here to stretch the strand. So these things happen. Now we have two almost straight, at least few portion of the DNA strand is almost straight. They are separated. Both the strands are separated from each other, right? Each strand work as a template strand, and that's the semi-conservative type. So basically, this is the template strand. So this strand will make one complementary copy, and this is another template strand which is going to make another complementary copy, and that's how we are going to get two new strands. Now, always this copying thing occurred from 5' to 3' direction. Okay, so basically this strand, it runs from this one, it runs from 5' to 3', and this runs from 3 to 5. Okay, so in this case, for this one, it would be completely direct for this one. Okay, so it will go for 5' to 3'. I hope you understand. So these two strands, when we are copying, so this is the template one, and this is the complementary strand. Complementary means if this strand's template goes from 5' to 3', then complementary must need to go from 3' to 5', right? Okay, so always DNA replication goes from 5' to 3'. Okay, so basically this is the direction, and we know that one old one means the template pairs up with a new one, and ultimately it produces two new strands. So that's the the big picture. E. coli has very small genome. It has 4.6 million base pairs. Okay, so if you go by one nucleotide, let's say this is adenosine, then A, T, G, C, if you count, there are 4.6 million base pairs are there inside E. coli chromosome. So you can understand. So there are 3 steps: initiation, elongation, and termination. So basically what happens? So first step was that these two strands, they get separated, okay, and now almost straight, okay. One enzyme, it's called DNA polymerase. Now DNA polymerase enzyme, it's DNA polymerase 3 enzyme. It's very unique, okay. It has unique characters because it can read the DNA sequence. Okay, so let's say over here it has A, then T, then C, then G, C, G. So the DNA polymerase can read that sequence. And at the same time, what it can do, it can pick up the complementary base pair over here at the same spot. So once it read A, it will bring the complementary one. When it read T, it will bring the complementary one. C, it will bring the complementary one. Okay, so that's how that DNA polymerase actually makes the new strand. Okay, so main catalytic enzyme for DNA replication is DNA polymerase 3. Very important enzyme. So it read and synthesize at the same time. Now there is a problem. Problem means that DNA polymerase very good, you know, it will read and it will synthesize, but it only can add nucleotide if there is one DNA or RNA template or RNA nucleotide is there to elongate. Okay, so if there is no DNA or RNA nucleotide available, then DNA polymerase cannot start the DNA replication. So basically the problem is, to start the DNA replication, we must have a small piece of DNA or RNA nucleotide. Okay, so if there is a small piece of DNA or RNA nucleotide is there, then and it has to be from 5' to 3', then the DNA nucleotide can add up rest of the DNA nucleotide from there, and this small piece is called primer. Okay, so that's the problem. So we must have a primer, small piece of primer that has a free 3' end. So I hope you remember the structure, right? So 1, 2, 3, 4. Over here we have 5, right? So over here at the 3 position we have 8. So DNA polymerase can only add nucleotides, means another one, only if it has that free OH end. Okay, so and this small piece is called the primer, which has that free OH end. Now DNA polymerase cannot make this one, this primer, it cannot make. So we have another one enzyme, it's called RNA polymerase enzyme. RNA polymerase, it can start replication from 0. It does not require any hanging OH situation. Okay, so what it does, it actually make a small RNA primer. So it is RNA polymerase, means it can catalyze RNA, so it actually make a small piece of RNA that has a free OH end. So once both the DNA strands get separated from each other, then RNA polymerase must need to make one RNA primer over here. So once that RNA primer is at position, then DNA polymerase, this enzyme, it can start adding up all the nucleotides. It can read all the sequence, and accordingly it can collect the complementary base pair over here, and that's how it can make the complementary DNA strand. Okay, it's very simple, but remember the question arises that these two strands are anti-parallel. So if it runs from 3' to 5', then only that new DNA strands could be made from 5' to 3', and that's the only sequence DNA polymerase can't, only direction DNA polymerase can't make a new strands. Always DNA replication goes only one direction, and it goes from 5' to 3'. Okay, why? Because remember that at the 3' we have that OH group and this 5, so it needs to at top. So 5, 3. So 5 will add up, 3 will be here. 5, 3, 5, 3. So basically that's how it goes. So from 5 to 3, it's always. Okay, so to synthesize that 5' to 3' complementary strand, the template strand must run from 3' to 5' direction, right? Because this is the complementary strand. Okay, now the question arises. So this strand, if this is one template strand, this is another template strand, right? So if it was 3 to 5', then of course it was 5 to 3'. So it was running from 5 to 3'. Now how it is going to make, because then it should be 3 to 5, right? The complementary strand, if it makes complementary strand, then it should go from 3 to 5, and that's not the direction. It should go on this way, right? Okay, so that's the problem over here, but we have solution too. So over here, what it does, for the other strands, basically instead of just one RNA primer, that RNA polymerase make multiple RNA primer. Okay, so it add multiple RNA primer, and instead of going from this direction to this direction, and it continuous, instead of continuous synthesis of one strand, it makes basically small pieces. Okay, so it will make, so this is the RNA primer, so it will make, first it will make this much, then it will jump, it will make another one over here. So basically it makes like that way. So for this strand, it's multiple small segments are there. Now what happened? We have another enzyme, DNA polymerase 1 enzyme. So it was DNA polymerase 3 who is actually going to synthesize. Now this is another one, DNA polymerase 1. What it does, when all these segments are made, it's done, then it actually clean the RNA primer. It takes away the RNA primer. So when it takes away the RNA primer, so what's the situation? So this is the template strand, okay? And one small piece, one small, right? Because it is going from 5 to 3, 5 to 3, 5 to 3, right? But there is a gap in between. Now we have another enzyme, it is called DNA ligase. That enzyme actually add up all the small pieces together, and that's how it actually make another complementary DNA strand. So what happened? All together, what happened? So DNA replication goes continuously for one strand. So this is continuous replication, but for the other strand, it is actually discontinuous. Okay, so that's the big picture. Basically, I talked about everything about DNA replication. Now, few more detail you need to know. So I hope you can understand that. So this was the DNA. Okay, so this was the DNA. So at certain point, it needs to open up, right? Like let's say it has to, DNA replication needs to start from here. So when it opens up, so basically it looks like this. Okay, now this is the starting point of DNA replication. That starting point is called origin of replication. So from where actually the DNA replication start, or you can say that helicase enzyme actually start breaking that hydrogen bonds. That point is called the origin of replication. So for us, for eukaryotic chromosome, we have a linear chromosome, right? It's a linear chromosome. For us, what happened? We have multiple origin of replication because it's really big. So if we start from one point, so to replicate the whole 6-meter long chromosome, and we have 46 chromosomes, it will take years for cells. So what cell does, it actually make multiple area. So maybe from here to here, they will it will open up, unwinding. Okay, so we have multiple origin of replication, but for the bacteria, they have one circular chromosome, right? So double-stranded. So for them, they just have one origin of replication, and the DNA replication goes bidirectional, both the direction it goes. So basically, they open up this way. Okay, so let's say it was like that way. So they open up, so this side goes this way, this side goes that way, and goes this way and goes that way. Ultimately, when they meet, so they stop replicating. So for bacterial, or you can say for prokaryotic DNA replication, it's bidirectional, but it has only one origin of replication, versus for eukaryotic, we have multiple origin of replication. So I already talked about all this enzyme. Okay, now when the chromosome, both the strands get separated from each other, what happens? It looks like some area looks like very thick, floppy area, and this region is called bubble. Under microscope it looks like bubble. Okay, so that's the replication bubble, and we have multiple of replication bubble. I think I do have, yeah, over here you can see. So this is a linear chromosome for eukaryotic, and this is the bubble. This is one bubble. So we have a multiple replication bubble because we have multiple origin of replication. Okay, so there are multiple replication sites along the length of the DNA for eukaryotes, but for the prokaryotic chromosome, as I told, that one origin of replication, so basically it goes both the direction, same process, and once they meet, they actually stop. Now the fork-like area, when the two strands get separated, that region just near the origin of replication, it looks like fork. Okay, so that area is called replication fork. So I told you that the one strand that runs from 3' to 5', that side, the DNA synthesis or DNA replication is continuous. Okay, so that strand is called leading strand. So the strand where the DNA replication goes continuously from 5' to 3' direction, that strand is called leading strand. But the other strand, the template strand, was running from 5' to 3'. Over here, they make small segment at a time, right? But still, this side, it goes from 5' to 3', right? So that's small. So this side, it makes small fragments at the beginning, and all the small fragments are called Okazaki fragment. Okay, scientist Okazaki first found out. So to show respect, we called all those segments as Okazaki fragments. So this strand is called lagging strand. So basically it lags behind the continuous strand. So that's why it's lagging strand. So DNA replication, in one strand it goes continuous, and that strand is called leading strand. other strand, it is discontinuous. It makes small Okazaki fragments, multiple small Okazaki fragments, and at the end, that DNA ligase actually connect all the small fragments together. That strand is called lagging strand. So DNA replication is discontinuous on lagging strand. But for both the strands, DNA replication goes from 5' to 3' direction. Okay, the direction is always same. To maintain that this strand, the DNA replication is discontinuous on for this strand. Very important to remember all these enzymes, name of the enzymes and what they do. Like DNA polymerase 1, it actually cleans the RNA primer. It picked up the RNA primer after the lagging strand synthesis, replication. Okay, ligase, it actually connects all the small pieces together. Topoisomerase 2, basically topoisomerase 2 and 4 helps to unwinding. That single-stranded binding protein, it actually stretch the DNA strand. And main enzyme that catalyze the DNA replication, that's the DNA polymerase 3. Okay, DNA polymerase 3. So it actually the who catalyze the DNA replication, plus it has another function. It's also works as a proof reader. Okay, this enzyme is responsible for proofreading also. Okay, so who is the main catalytic enzyme for the DNA replication? DNA polymerase 3. Okay, and make sure the DNA polymerase 1, what it does, it actually pick up or remove that RNA primer. So that's the difference between DNA polymerase 1 and 3. So helicase, unzipping the DNA helix, how it unzip? It actually breaks the hydrogen bond. Okay, so I talked everything about it. Okay, telomerase. So telomere, the word telomere means the end of the, so basically both the ends of chromosome is called telomere. Okay, now you can understand that chromosome has so many important coding region. Okay, so if it breaks, then we are going to, so during replication it can break, then we are going to lose all the genetic information. So to avoid that, we have telomerase enzyme. So telomerase enzyme, what it actually does, so at the end of chromosome, so let's say this is the chromosome and this end is called the telomere. Okay, so to protect all the coding sequence, coding region, both the ends are secured with some a non-coding sequence, you can say non-coding sequence, and it goes T T A G G G. This is the sequence. Okay, this small 6 nucleotide sequence. They repeat 100 to 1,000 times at both the end, this area and this area. Okay, now this telomere is in, so if it breaks, then it breaks the non-coding sequence. We are not actually going to lose any coding sequence. That's the purpose of having this telomere part, means the end part of the chromosome. Okay. Now, one enzyme, it's called telomerase enzyme. It actually has it has two functions. Okay, it actually has one catalytic part, catalytic function, and another function is it actually provides one RNA primer. So let's say every time when DNA replicates, you can understand that when DNA is replicating, okay, so that RNA polymerase is moving, okay, so it started from here and it is going. So ultimately, let's say when it is here, when it reaches here, there is no space to read this much portion. So it actually stops here because that DNA polymerase cannot fit within this period. And ultimately, once this DNA is going to replicate, it is going to lose this portion of the chromosome. So every time it happens, every time when DNA replicates, we are going to lose a little bit of that telomere part, means the end part of the chromosome. So to protect that, this telomerase enzyme, what it does, like let's say this is the chromosome, okay, at 3' end, what it does, so it has primer, so it provides the primer part. So it actually make one complementary RNA part. This is RNA. And this is the DNA, so complementary. You can see GCTA, so that's a complementary part. Okay, so it provides that complementary part, and it has another function, that catalytic. Then it actually add the rest of the part of the the DNA. So it actually make. So basically, that's how this telomerase enzyme it tries to rebuild the lost portion. Still, every time we are going to lose the little portion of the telomere, and that's the reason now scientists, they believe that that's the reason we get older. Okay, our age actually comes because of this reason. But this telomerase enzyme, like which is actually going to rebuild, adult people do not have that telomerase enzyme. So that's the reason we are constantly going to lose that end portion of the chromosome, and that's how we are getting old. That's the reason behind of our age. But inside the germ cell, that we can find out, and the stem cells, we can find out the telomerase enzymes. But adult people, we don't have telomerase enzymes to protect or to rebuild the telomere part. Okay, so this type of math you can expect. So Clostridium tetani DNA is 14% guanine. What percent of his DNA is adenine? Okay, so we know adenine goes with thymine, cytosine goes with guanine, right? So if we have 14%, so basically these are the strand. If two strand, if we have adenine over here, then we have thymine over here, right? So how many adenine is there? Exact number of thymine is there. How many cytosine is there? Exact number of guanine is there, right? So if we have 14% guanine, then we have 14% cytosine, right? So these two together gives us 28%. Okay, so 100 minus 28%, 100% minus 20%, that gives us 72%, means this is the percentage for adenine and thymine. So if together they make the 72%, then A 36%, thymine 36%. So this is the right answer. Okay, now the transcription. Um, so transcription, remember, transcription means when we make one messenger RNA, or you can say RNA copy from DNA. So that's the process of transcription. Over here again we have three steps: initiation, elongation, and termination. Transcription always goes from 5' to 3' direction, exactly like DNA replication. It's not that much complicated like DNA replication. Okay, so what happens? So in this case, if this is the DNA template, so let's say these two are the DNA template. Okay, replication, for replication, the entire length of chromosome is going to be copied. Okay, but for transcription we really do not need to copy the entire length of chromosome. Remember, the purpose of transcription is to make protein. So if that's the purpose, we only copy the portion where we have that gene that actually codes for that protein. Like at this moment, let's say my body needs insulin, and this is the portion where I have that insulin gene. So the entire portion of the chromosome will be like that, but only this portion will get separated from each other, and this portion is only going to be transcripted. So unlike DNA replication, that a transcription occurs only a very small portion of the DNA. Okay, and that part or of the chromosome work as a template or antisense strand. Okay, it occurs 5' to 3' direction. Now it's pretty easy. So exact same way over here we are making actually RNA from DNA, right? So for DNA replication we were making DNA to DNA, right? So that's why our enzyme was DNA polymerase. Over here we are making RNA, so the main catalytic enzyme is RNA polymerase. So that same polymerase enzyme, it can make any strands from that goes from 5' to 3' direction. But in this case, it is RNA polymerase. So we really do not need any RNA primer. We do not need that. Okay. Now, over here, what happens? So it starts that transcription start from a region that is called promoter. That's the region it start, and okay, and at the end there is a region, it's called terminator, where that DNA replication stop, DNA transcription stop. Now, what is promoter region? Promoter region means there is a specific sequence of nucleotide. Terminator regions means specific sequence. Maybe it could be like promoter, let's say it's TATA, Tata box. So if this is the sequence TATA, so cell knows that RNA polymerase can recognize that that's the promoter region. So it will sit over here and it will start the transcription. And exactly same way for terminator, it has another specific sequence. Okay, so when that RNA polymerase will reach that side, it can recognize that it has to stop, then it stops. So initiation, so when that RNA polymerase sits on the promoter region. That's the initiation. So it sits over here at the promoter region and it start reading the sequence. Okay. Then the second step is elongation or elongation. This time what it does, that RNA polymerase, it start reading the nucleotide sequence and it pick up on the RNA nucleotide. Okay, and it actually elongate the length. So that's the elongation. Termination, when that RNA polymerase reached at the terminator region, it stopped. So it's pretty simple, that transcription. Template strand is called the antisense strand. So remember, both two strands are there. Okay, so if this is the template strand, then it's called antisense strand, and the complementary strand, that is called sense strand. Positive, it's actually called positive sense strand. Okay, so by the process of transcription, cell makes that messenger RNA. Okay, for eukaryotic chromosome, what happens? Remember, inside the eukaryotic chromosome, we have both coding sequence and non-coding sequence. Okay, and it is very costly. So inside the nucleus, this was the DNA, and cell makes that messenger RNA. So DNA has both coding and non-coding sequence, and that RNA messenger RNA has an exact copy, so it carry both coding and non-coding sequence. And it will come out of the nucleus all the way to the ribosome, and ribosome will read all these. It will take time to read, to go over all these, plus it costs energy. Okay, to save that time and energy, what actually eukaryotic cell does before that messenger RNA, before it comes out of the nucleus, it actually takes out all the non-coding sequence. Okay, so first it cuts out. So if this is the messenger RNA, let's say this is the non-coding sequence. Okay, this is the non-coding sequence, and this is the coding sequence. Let's say this is the non-coding sequence. So it first it makes small pieces of all those, then it add actually all the coding sequence together. Okay, and this process is called RNA splicing. So non-coding sequence are called introns, means interrupting sequence, and all the coding sequences are called exons. So during RNA splicing, cells removes all the introns, and it keeps all the exon sequence connected to save energy and the time. So that is one thing happens, that eukaryotic processing. So once that RNA splicing is done over here, then that messenger RNA. So at the beginning, the cell makes really large, huge messenger RNA. Then after RNA splicing, it's a very small piece of messenger RNA comes out of the nucleus. So that's one eukaryotic cell processing. Other thing happens, so when messenger RNA comes out of the nucleus, look, it's very valuable. You know, you can understand that it's carrying all the sequence genetic information. So what it does, it actually add a cap at the 5' cap means so methyl group. It adds multiple methyl group at the 5' end, and multiple adenine sequence. So it's called poly A tail. So it's a tail. So basically it add one head portion and one tail portion. So tail goes at 3' end and head goes at 5'. Head means it's multiple CH3 methylene group, and tail portion goes multiple adenine. So 5' cap and 3' till they add. This is the RNA splicing. Okay. Next, the genetic code. This chapter is really big. Genetic code, it is the relationship between the mRNA codon and its corresponding amino acids. Okay. So what happens? So that mRNA, okay, it is carrying all the coding sequence. Okay. And remember that when it was on DNA, so all these are the nucleotide sequence: adenine, cytosine, adenine, guanine, thymine, right? So when it is messenger RNA, during transcription, remember that RNA, it does not have thymine. Thymine is replaced by the uracil, right? So over here, and it's complementary of the actual DNA strand. So over here, this is the original DNA strand, okay? So first nucleotide on DNA, it was adenine, okay? Then the complementary strand for DNA should be thymine, right? It should T, but for the messenger RNA, for the RNA does not have T, so it should be uracil. So uracil. So it was C, complementary strand should carry guanine. C, guanine, adenine, then again uracil, uracil, uracil. So it goes like that. So that's the complementary messenger RNA or complementary RNA strand. Okay. So make sure you can understand this part. For test, I can give you. So if I give you, let's say this is 5' and this is 3'. This is DNA strand, okay? I have adenine, cytosine, guanine, adenine, guanine, guanine, guanine. If this is the combination, what would be your messenger RNA strand? Remember, first thing, it has to be complementary, right? The messenger RNA. So that messenger RNA should go from 3' to 5'. Okay. Now write down the complementary. So adenine. So instead of thymine, it should be uracil, then guanine, then cytosine, uracil, cytosine, cytosine, cytosine. Make sure you understand. You can expect at least one question, and don't forget the direction. So if this is the DNA, okay, if I ask you to make complementary DNA, so this is DNA, okay? If I ask you to make complementary DNA strand, then again complementary means it will be 5'. So this is DNA. We are making DNA. So adenine means we have thymine, guanine, cytosine, thymine, cytosine, cytosine, cytosine. But this is RNA. Okay. Now on RNA, we actually read, or cell actually read 3 nucleotide together. So 3 nucleotide together is called codon, and all this codon represent one amino acid. Okay, so the relationship of mRNA and the meaning of this codon is actually the genetic code. Okay, like UCG, this codon, this is one codon. So this is one codon, these 3 together, second codon, this is the third codon. So over here, these 3 should be one codon. Okay, then these 3 should be one codon. So it goes like that. Okay. All these codon represent amino acid, and you can find which codon represent which amino acid. You can find that information inside the genetic code. So that's the relationship between the messenger RNA and the amino acids. Okay. Inside our cell, inside our body, we have 20 predisposed amino acids. 20 different types of amino acids are there, like trypsin, one amino acid, phenylalanine, that is another amino acid, glycine, another amino acid, serine, another amino acid. So over here we can see 4. So we have 20 different types of amino acids already inside our body. So this is the genetic code you can see. So if the messenger RNA carry the codon UUU, that represent phenylalanine. If it is CUU, that's the leucine. If it is AUG, that's the methionine. Okay. Now AUG, that's our start codon. Protein synthesis start with this codon. Okay, to start the protein synthesis on messenger RNA, we must have this AUG. So if this is the messenger RNA, our first codon should be AUG. Okay, so let's say this is the messenger RNA. Now the ribosome, once it will see that, okay, so this is the start codon, then it will start protein synthesis from here instead of here. So that's the start codon. We have 3 stop codon: UAA, University of Alabama, University of Augusta, and University of Georgia. These are the 3 stop codon. So protein synthesis will stop if one of these 3 stop codon are here. So if you have University of Alabama over here, ribosome will stop protein synthesis over here. Make sure to remember that you have to memorize the stop codon and the start codon. So all together we have 64 codon, okay? Because we have 4 nucleotide, right? The adenine, guanine, cytosine, uracil, and the genetic code actually goes by the messenger RNA, goes by the RNA. So we don't see thymine over here, we see all uracil. So 4 over here, 4 nucleotide, and we have 3 codon, the 3 base, right? 3 base together make 1 codon. So 4 into 4 into 4. That gives us 64 amino acids, 64 codons, 64 different types of amino acid. You can see, look, that for glycine, we have 4 different codons. So there are amino acids, because you can understand we have only 20 amino acids, right? So for one amino acid, there must be more than 1 codon, because we have 64 codon, right? So out of 64, these 3 stop codon does not make any protein. They don't represent any amino acids. So for 61 codons, we have all amino acids. So 61 codon represents amino acids. Okay, so we have 20 amino acids which are represented by these 61 codons, because the start codon actually represent amino acids, but the stop codon, they don't. So genetic code is redundant, means with more than 1 codon for some amino acids. Unambiguous, in that any codon for one amino acid does not code for any other amino acid. Okay, so if it is AGU, it has to be serine. Okay, for serine we have 2 codon, but every time to make serine, one of these 2 codons should be there. Universal, from bacteria to human, everybody follow the same genetic code. Protein synthesis occurs in ribosome. Okay, so ribosome has 2 units. One is large subunits and one is small subunits. For eukaryotes, the large subunit is called 50S, small subunit called 40S. Okay, but for the prokaryotes, it is 40 and 30. So 2 parts, and rRNA and the protein together, they make the ribosome. Now another ribosome, so you already learned the structure of a messenger RNA. This is the rRNA that makes the ribosome. Now the tRNA, transfer RNA, it looks like this, okay? At the head part, it has a one head part and one tail, okay? Head part, it carry one amino acid. It's transfer RNA, it transfers something, right? So what it transfer, it actually transfer amino acids. Okay, so at the head part, so it carry that amino acid. Out of 20, it carries just one amino acids. Now at the leg part, there are 3 nitrogenous bases are there. These 3 nitrogenous bases are called anticodon. Okay, so why it's anticodon? So we can find codon on messenger RNA, right? Okay, so let's say if it is EU, A, C, if this is the codon on messenger RNA, then on tRNA, it should be A, U, G. Okay, so it's complementary of the messenger RNA. So that's why it is called anticodon. So codon we can find on messenger RNA. Anticodon that's on tRNA. Okay. This is the tRNA. So this is the head part where it actually carries the amino acids, and this is the leg part where it has that anticodon. Now the protein synthesis, how actually protein synthesis happens, okay? So this is the ribosome. So ribosome, it has 3 sides. This is the large subunit, and this is the small subunit. It has 3 sides. One side is called A, one side is called P, and the another side is called E. Okay. Now what happened? The messenger RNA actually come and sits at the junction of both the subunits. Okay, it sits over here. Now ribosome will start reading, and all those tRNA, so this is the anticodon, and they are carrying different amino acids. So 20 different types of tRNA are there. Okay, so 20 different types of amino acids are there. They are constantly moving, they are passing by this area. Okay, so rRNA, they did their work. They made the ribosome just immediately before the protein synthesis. Now the messenger RNA come and get stuck at the junction between the large subunit and small subunit. Now ribosome will read. So let's say we have lots of C over here. That's our start codon. Okay, so these are the defined codon. So this is codon 2, this is codon 3, like that. So this is our start codon. So protein synthesis will start from here. So that part, that start codon area, will come and sit at the A side. Once that AUG comes over here, so let's say now so basically the messenger RNA moves from this side to this side. So this is moving this side. Now over here, it is actually that start codon sits over here. So AUG, okay, that's the start codon. Now, once it's here, ribosome understand that's the start codon. Okay. Now, all the different tRNA, they are carrying so many different type of amino acids, right? So all these tRNA, they are moving around this area, but only the amino acid that has anticodon that will match with this codon. So A means EU, should be EU, A, C. So that tRNA whose anticodon will be complementary with the start codon, that can come and sit over here. And this tRNA, so this anticodon actually, this tRNA carry the amino acid that correspond with this anticodon. Okay, so let's see that AUG. Okay, that's the amino acid, so methionine. So basically over here, this tRNA will carry that methionine amino acid. Okay, now once this codon and the anticodon fit, then that whole messenger RNA, the whole thing, that the tRNA and the mRNA will move one side. So it will now come over here, this side, P side. Once that mRNA tRNA combination comes at the P side, then it will release that amino acids. Okay, now it will move one more time, it will come to the exit side. Now this tRNA is empty, it already gave that amino acid to the ribosome. It does not have any amino acid, so it will take exit from this side, and it will go to grab another methionine amino acid from the cell. Now the second codon will sit over here. Another tRNA corresponding tRNA will sit over here. They will move over here. They will release another amino acids over here. They will move this side and they will exit away. Now the third one will come. Then another tRNA with corresponding amino acid will sit. Then they will move over here. It will release the amino acids. Okay, so basically that's how it's make that polypeptide, and then ribosome will make peptide bond in between the amino acids, and that's how it makes protein, right? So all the amino acids, peptide bond, that gives us polypeptide chain. So that's how cell makes protein synthesis. So again, 3 steps: initiation, elongation, and termination. Initiation start when that start codon sits at the A site. Okay, elongation, the middle part, it continuously, that messenger RNA and tRNA combination occurs at the E site. They move from A site to P site, they release the amino acids and they move to the E site. That's the elongation side. And then when there is a stop codon, like let's say University of Georgia, okay, once that this stop codon comes over here, then ribosome does not allow any tRNA to sit because it does not have any corresponding amino acids. Okay, then it will messenger RNA will be separated from the ribosome. Okay, and the protein synthesis will stop. So that's the process of translation. Okay, so the last part, the mutation. Okay, mutations means change in nucleotide sequence. So let's go back to the genetic code, so you will get it. Okay, I forgot to tell you one thing you really need to understand. So if I give you any DNA sequence, let's say I gave you AAA, then let's say C, G, T, and then G, G, C. If this is the DNA and I ask you to make protein, so what protein it is going to make from this DNA? So this is our DNA sequence. Okay. So what we have to do to make protein? First, we have to make messenger RNA, right? So we have to first make messenger RNA. So AAA, complementary strand. So we don't have, RNA does not have T, so it should be EU, EU, EU. That's the first codon. Then C is G, C, thymine means it will make adenine. Then C, C, G, right? So that's the transcription part is done. So from here to here, now the translation part, protein part. So we know the 3 amino acids are there. So U, U, U. What is U, U, U? U, U means phenyl. So that's our first amino acid, phenylalanine. Then GCA. So G, then CA, alanine. So second amino acid is alanine. Third amino acid CCG. C, this is C. Oh my. CCG, that's the proline. So our third one is proline. You can see here. Okay. So that's how we actually can tell that which amino acid cell is going to make. Now, oh, sorry, I went over there to talk about the mutation. So, look over here. Let me change the color of the pen. So, you can see over here when DNA is replicating. So, complementary strand should be T, 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 right? Then this should be C, G, C, A. Mutation means the change in the nucleotide sequence. So let's say instead of TTA, cell forget to transfer A to thymine if it keeps A. So TTT is going to produce phenylalanine, right? The original, based on original coding, cell is supposed to make this, but now TTA. So TTA means basically EU. So if it converts into, so EU, what I did wrong? EU. Okay, let's go for this one, GCA. So instead of A, so basically it was supposed to be CGT. So this is the original. So it should make GCA. So if instead of this it gives C, means over here it should be G. So GCG, GCG. So it should be alanine. So in this case, it's fine, the same. But instead of alanine, if it makes something else, then completely different protein is going to make. Okay, instead of black hair, I would have brown hair or gold hair, blonde hair. Okay, so that's a mutation. Okay, so and mutation has to be heritable. Okay, so any heritable change is called the mutation. Okay, mutations in body cells cannot be passed on to your children. So if you have mutations on your cell, like when you are standing for a long time under sun, mutation can happen. Okay, any nucleotide sequence can be changed because of the UV ray, but that won't be heritable, because you cannot pass your body salts to your children. But if it is in the sex cells, sperm cells or egg cells, then that can pass to the children. Now, so I gave you an example over here. So let me move this one. So original word was beast, B-E-A-S-T. Now B is replaced or substituted with the letter F. So now the new word is beast. Okay, so it's completely different meaning. Now insertion, another thing can happen. In between B and E, there is a new nucleotide base, that's R. That instead of beast, the new word is breast. Okay, now deletion. Okay, so B-E-A, A got deleted. Now it's best. Inversion, S and T changed their position, but it gives you a new word. So that's the mutation. Now mutation can be spontaneous mutation, like basically mutation always occurs in the DNA. Okay, so when DNA replicates, so basically during the DNA replication time, the DNA polymerase can misread or misprove read. So that can happen. That's a spontaneous mutation. Induced mutation, because of the results from the exposure, like UV rays. You are standing under UV rays for a long time. That can brings the mutation. Any poison, any gas, all those things. So um, all these things, the environmental factor that can induce mutation, those are called mutagens. So because of the mutagens, mutation occurs, then that mutation is called induced mutation. Point mutation, that only changes occurs in only one nucleotide base. Okay, so over here B got substituted with F, so just only one nucleotide base has been changed. So that's the point mutation. So just one point of the chromosome got changed. And it can happen with addition of nucleotide, deletion, or substitution of single base, just one nucleotide base. So AUG, that's the start codon, methionine. Okay, the U got replaced with A, substituted with A. So instead of methionine, cell is going to make lysine. So that sickle cell anemia. Sickle cell anemia, what happens? So the normal protein, that's the hemoglobin, that actually helps to carry oxygen throughout the body. Okay, so in case of normal hemoglobin, that valine, the protein valine, got replaced with glutamine. Okay, so instead of valine, cell produces glutamine. So, and that's the result. The normal hemoglobin become D-shaped. Okay, so it got like crescent shaped. Okay, and it cannot carry that oxygen because of the shape. Because instead of valine, we have glutamine, and that completely changed the shape of the hemoglobin protein. Now this new protein is hemoglobin S, means sickle cell hemoglobin. And one hemoglobin can carry multiple oxygen. Now they cannot carry oxygen. That's how that sickle cell anemia occurs. So you can understand. So over here, if start codon, this is the start codon. Start codon convert into completely different codon, then that protein synthesis will not occur. So I need insulin Okay, at this moment, my glucose level is so high. I need insulin. Body needs to make insulin. But what happened? I have a mutation at my start codon. Body cannot even start the process. Because if start codon is not there, then protein synthesis will not start. Just only change over here can stop the entire process. It's a huge, huge loss for the cell. So you can understand the effect of the mutation. Addition in insertion. So instead of AUG, one U got inserted. Deletion, that U got deleted. So again, that that start codon got completely changed. So entire process of cell protein synthesis will stop. So the point mutation, there are different type of point mutation. One is called silent, has no effect on the protein synthesis. Like I gave you example, like over here, let's say the T, it was supposed to be A. Okay, so A got changed into T, but ultimate product, because for valine, we have multiple codon, right? So there is a mutation, but it really does not have any effect because the same protein is going to produce. So that's called silent. Missense result in an amino acid substitution. Over here, instead of proline, it made thrombin. So it has a effect, bad effect. Nonsense substitutes a stop codon for an amino acid. So that maybe 100 amino acids are supposed to make, okay, the cell. But because of the mutation over here, the C got changed with G, and that makes a stop sign, stop codon. So after making only these 3 amino acids, then cell will stop making protein synthesis. Okay, so that if it is insulin, cell cannot make that insulin. Okay, so that's the nonsense. Insertion or deletion results in shift in the reading frame. Okay, so all this can happen, all this that point mutation can happen if it is substitution. So you can see that one codon got substituted with another one. So that's the point mutation, and there are 3 different types of point mutation. If insertion or deletion occurs, so what happened? The entire reading frame get changed. Okay, so I can show you the over here. The actual sentence was, the fat cat ate the rat. So it makes sense. So this is one codon, this is one codon, this is one codon, you know. So it makes a sense. You can understand the meaning of the sentence. Now over here, before C, 8 got inserted, 1 nucleotide base, new nucleotide base. Now the cell will start reading, it will go by 3, right? So the fat, then it will not make any sense. It will not represent any codon. The cell cannot make any amino acids. Okay, so that is called frame shift mutation, which alters the reading frame. So entire reading frame has been changed over here, and it will not make any sense to cell. Deletion, so the fat cat ate the rat, E got deleted. So then cell will read 3 at a time, right? So TEF, ATC is a tape, then it cannot read. It will not make any sense. So that's the frame shift mutation. So 2 types of mutation, point and frame shift. Point mutation caused, the reason of point mutation is substitution of nucleotide base. When insertion or deletion occurs, then that gives us frame shift mutation. Make sure to know, so ATG, G got substituted, so that's the point mutation. The dog bit the cat. The dog beat the car. So T got substituted with R. Again, this one is point mutation. So you can read all the way, but only 1 point it has been changed. The dog beat the cat. So enter reading frame has been changed. Then this is the frame shift mutation. Okay, that's all today. Thank you for watching.
Thank you, Miss Nandi.
Oh, you all are here still?
Yeah, we're here. That means two of us.
Okay. I thought I'm the only one. Okay. So
thank you.
Yeah, read, and I posted the schedule. Okay, so we will review. I think towards the end, before third exam, we are going to review.
Okay.
Awesome. Thank you.
Sounds good.
Okay. Have a good night.
You too.
Good night.
Bye.